From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 31, recorded on June 27th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hey, Vincent. Hey, everyone.
And I presume you're no longer skiing, right?
No longer skiing, no. Summer is here, in the 90s, in the valley, at least.
Also joining us from New York, Tim Chung.
Hello. Hi, everyone. Good to be back. Not skiing here either.
No, we're not skiing here. Although, over in the Meadowlands, there's a indoor ski slope that they built.
Is it fake snow or is it?
I think it's
fake.
It's
got to be fake snow.
Passed it yesterday.
Okay.
You could see it from the turnpike. It's like, gosh. And from her new location in New Orleans, Vivian Morrison.
Yay. Hello, everybody. It's Mardi Gras here all the time.
So you made the move and you're at Tulane University in New Orleans and everything good?
Yeah, everything is great. I'm super excited.
So everybody's there. You, the husband, and the kids. Everyone's together, correct?
Oh, yes, we are. And we were very much together last week when, after 2 days of being in summer camp, my daughter tested positive for COVID. So then we were just right back in our house together. So I'm just keeping my fingers crossed that we don't have a rerun of that this week.
Kids are okay with New Orleans?
Yeah, they love it. They love our new house. We all love our new house and the location, and they're just, happy as clams.
Good. Glad to hear it.
I like the blinds. Look very nice.
Very nice.
This house is amazing. It's like, it's kind of like Breckenridge, but in New Orleans. It's like lots of wood, and so it's a one-of-a-kind place.
That is not what I would have pictured New Orleans. Breckenridge.
I know. I mean, it's like a cabin on the inside, but it makes it feel really homey.
Cool. All right. Well, enjoy your new digs.
Thanks.
And meanwhile, for your neuroscience education today, Jason is going to lead us.
I am. And this is a paper that probably needs a fair amount of context, and we'll go through that before we actually go through the paper. But the paper is called Ccr5 Closes the Temporal Window for Memory Linking. And this comes from the main group is Alcino Silva's group at UCLA. And there's a lot to unpack here. So temporal window for memory linking. So what does that mean? So, episodic memory, which is what we, mostly think about memory, which is our experiences during the day. So if I asked you, what did you have for breakfast? And then say, well, who else was with you at breakfast? And I ask you that now, and then maybe an hour later, I'll go, well, what did you do after breakfast? Do you remember what you did after breakfast? And so there's a temporal order. And so obviously most of us could answer that and say, I had, raisin bran, and then I went to work. And if your memory is working correctly, you can link those things together. So those are temporally easy because they're one thing after the other. But if I say to you, well, what did you have breakfast a week ago, and what did you do after that? Unless it's something that's very normal, so like, I had breakfast, then I went to work again. That it can be hard to recall something that's temporally linked, that's far in time. But so there seems to be a way for the memories that when we're recording them to know that temporal link, so that things don't get jumbled. So if you recall breakfast from last week, it's not then associated with something that you did yesterday. And we know that many people do have issues with the sort of temporal linking, especially with cognitive aging, dementia. But even in young adults where you have psychiatric disorders, one of the first symptoms for schizophrenia is this temporal disorder of recalling events in your life. And, they can manifest as things like hallucinations, hearing voices. And that's partly because there's this temporal disconnect between what you're experiencing right now in the present versus what was you experienced a few days ago or whatever. And so obviously, if you're trying to understand memory, you would love to know how does the brain link things together so that there is a temporal order, and then what's the mechanism of keeping those things separate, as in we can't link everything together, because if you did that, then you would also have an issue. And so that's sort of the main question of this paper, but it's really delving into it at a very molecular and cellular level to say, in the brain, what are the cellular molecular mechanisms that help link memories together, and then how do you separate events? I just want to quickly comment, like this memory linking thing, I kind of taking the ability to separate different memories into different chunks. I kind of take it for granted. So I can remember what I ate for breakfast today and what I ate for breakfast yesterday, where it's kind of similar thing. It's just zero. But like what I ate for lunch yesterday and what I ate for lunch 2 days ago, I can kind of remember those two separately. But one obvious time when my memory linking got all jumbled up was during the pandemic, when I was doing pretty much the same thing every day and I was just staying at home, and that just became one big block. And I can't remember what I did during then at all. So yeah, so it's actually...
I think if you do something different, you remember that. But if you do the same thing every day, well, that's easy, right?
And if I'm staying at home every day, and it's just the same environment every single day, then everything just got jumbled up, and I can't tell you what I did, except for listening to podcasts, I guess.
By the way, Tim, can you lower your laptop a bit, get less headroom?
Oh,
tilt it down. Yeah. Now we see Wemu. Very good. So now I know what Jason eats for breakfast.
Raisin Bran?
Yeah,
Raisin Bran, well.
Jason, so you were saying that in schizophrenia, there's this this development of a deficit in ordering memories and things like that. Is the flip side, or potentially like the individuals who have photographic memory that can remember everything that happened on a specific day that was decades before? Do you think that those are kind of two ends of the spectrum, or do you think that they're really standalone?
Yeah, no, it's an interesting question, because there are these groups of people that seem to have photographic memory. And then the question is does that hinder them in some way? If you could remember every single thing that happened to you every single day, would you be able to then assign significance to something that should be assigned significance versus just something that you always do? Like, I put a t-shirt on versus I got married yesterday. And I mean, it seems like most of those people function just normally. They just seem to have this really specific domain that they can remember things, but it's not generalizable to everything. So it seems to be most of them have a photographic way of memorizing things that are like domain specific, like numbers or a calendar, or some have conversations that they can remember every constant. So it's not always. So it seems to be a fairly domain specific memory, but and then they seem to be functioning normally in society. It's not like that. So I think on that side of things, it's not a huge detriment to have a very good memory, or things that where you can actually recall esoteric facts or things that happen to you.
But I wonder if those photographic memory people is it a genetic component to it?
Yeah, maybe they lack CCR5.
Exactly.
It could be.
Yeah, I was like, let's link this to our paper in some way. Bring it back.
Yeah, it could be. I think there's like everything. There's probably some genetic component to this, and these are pretty rare folks. Like I remember UC Irvine had a study where they were trying to collect people who, claimed to have photographic memory, and of course, only a few of them actually did. But as far as I know, they haven't been able to figure out what's special about them, even at a sort of, when they would put them through an MRI scanner and try and figure out, is there anything grossly different in the brain? So back to the paper. This paper, of course, is not in humans. It's in animals. It's in mice, as most of our basic biology studies are. So the other sort of aspect to unpack here is Ccr5. So what is that? So Ccr5 is a receptor. So it's on the cell surface of a cell, and this is actually an interesting receptor that was studied in the context of infectious disease, so specifically HIV. So this is now, Vincent probably knows more about Ccr5 than I do. And it's a co-receptor for HIV. So if you the way the infectious particles can infect cells is that they bind to specific receptors on cells, and that allows them to then attach to them and get inside. And so then you're thinking, well, this is in the brain, so why would you have this receptor in the brain? And this is actually pretty common, that the evolution has reused receptors all over the body, and the immune system and the brain are the two most common places for receptors to be expressed. And historically, something is called a neurotransmitter if it was first found in the brain, and called a chemokine if it was a signaling molecule in the immune system. But that's kind of historical. So many of these chemokines and neurotransmitters you can find both outside the brain, inside the brain. So this is fairly common. But Tim actually did some research on trying to figure out why this group actually decided to study this particular receptor. So Tim, you can fill us in there.
Okay, so well, first of all, this has headline newsworthy clinical implication, because Ccr5 is actually the gene that the CRISPR baby got knocked out on. So I don't know if you guys remembered 2 years ago, maybe 3 years ago, a scientist in China CRISPR'd out the Ccr5 receptor in a couple of babies so that they are less susceptible to HIV. And then the scientist subsequently was put in jail because of massive ethical breach. So it's important to find out what Ccr5 is used for, not just in immune system, but apparently in in the nervous system as well.
But I looked pointing out that some people, about 10% of the human populations, lack Ccr5, and so we know that you can live without it, and if you delete it, you can be resistant to HIV. That's why he did that, because I believe there were twins born to HIV positive people, parents.
Yeah,
right?
Yeah. That's a very good point. I think that, it's also a naturally occurring mutation as well. But I think this group kind of stumbled upon Ccr5 as an important gene target for memory because they did a genetic screen. So they ordered many, many different lines of mutant mice from Jackson Laboratory, which is a supplier for mice. And then they just screened these mice for changes in memory. So the test that they used was they would put mouse in a novel context and then gave them a mild electric shock on the pores. And then this is called contextual fear conditioning, which Jason, this is going to be the bread and butter of the paper that we talk about today.
Yeah.
And then they later put the same mouse back into the same context after a long delay. And a mouse normally would remember that the context has previously been they got shocked in, so they would freeze. But if you wait long enough time, the mice would kind of forget and they would no longer exhibit this freezing behavior. But certain mice with certain genetic mutation would still exhibit this freezing behavior. And these group found that one of them is Ccr5. And apparently quite a few chemokines came up, and it suggests that chemokines are also important for memory, which is quite unexpected.
Yeah, exactly. So the other reason I think why they were looking at this particular receptor is that there was some previously published data suggesting that Ccr5 can negatively regulate an important gene for memory, and that gene is called CREB. So CREB is a transcription factor that is induced by learning and is super critical for memory formation. You take it out of mice or any animal, including flies, it's conserved between flies and mice, you don't get memory consolidation. So the hypothesis that they came in with in the start of the study was to say that Ccr5 activation would actually inhibit this gene expression program, and so would prevent or reduce consolidation of memory. The other thing that they then wanted to know was, is this receptor actually expressed in the parts of the brain that are important for making new memories? And that structure, the hippocampus, which we've discussed before, does express Ccr5 and CCL5, which is the ligand, and the receptor. So, you need both the receptor and the ligand for it to be activated. But what was interesting is that what they observed is you put the mouse in this contextual training. So you pair a shock with the particular context in the box. And so these are literally boxes where there's visual cues, there's auditory cues, and there's sometimes even smells that you can put in there to sort of change the experience of the animal. And then you pair that particular context with the shock, a mild shock. And the readout is freezing. So if the animal remembers that it got a shock in that particular context, as soon as you put it in that box, it freezes. And so it's a fairly easy readout because you can automate this, and it's it's high throughput. So this is a very common memory paradigm that I think we've probably just talked about before, but many labs use this. So after this training, the expression of Ccr5 slowly ramps up. And so by 6 hours after the training experience, there's an increase in the expression of Ccr5 in the hippocampus. But what was then even more interesting is that, and we've talked about this before, but now we have ways of tagging the cells that are active during that training period. And that circuit is called the engram, or the sort of physical circuit that we think is important for that particular memory. And so when the authors did this, they were able to show that the Ccr5 increase is actually in neurons that are predominantly in the engram. So suggesting that somehow the expression could be tied to memory. And there's actually this is a Nature paper, and it's extremely dense in data. There's something like 10 supplemental figures, but they introduce a lot of interesting modern neuroscience techniques that some of them we've covered, and this one particular one that they'll talk about later is optogenetics, where you can activate specific neurons with this light activated channel. But the something else that they've done as well is to actually engineer the Ccr5 receptor ligand interaction to be inducible by light. So instead of just having a channel that's letting in calcium, this is actually trying to mimic if you could activate the Ccr5 exogenously with light. So they're going to try and inhibit activity of Ccr5, and then they're also going to use ways to activate the channel itself without without generally activating the whole cell.
Jason?
Yeah.
So is the ligand to CCL5 at all involved in the optogenetic activation of CCR5, or you don't really need it at all in their paradigm?
So what they've done is just engineer the
like a change in CCR5 conformation or something like that when it gets exposed to light.
They've basically engineered the Ccr5 to be light inducible. Is one way to.
It's a chimera of two proteins, so it's kind of like CAR T cell therapy. But they put in a light sensitive outside domains that faces the outside of the cell that you can, when you shine a light, it causes a conformation change. And then on the inside of the cell, they put in the Ccr5 signaling regions, so domains, so that it talks to the right people. Not people, but molecules.
Right. One of Tim's classic metaphor or, examples.
It's all people inside the neuron.
It's all people.
But so that you can actually control you can use light to really temporally control when you activate the CCR5, and also you can put it in the cells that you want, which is neurons in this case.
And endogenous CCL5 is not a worry for them, or have they?
That's it probably isn't, because the outside domain is now a light sensitive rhodopsin. So it's similar to what is in your eyes.
So they don't actually have any endogenous CCR5.
No, they do, but
they do.
The light won't activate the endogenous CCR5, just this exogenous.
So it's a layering above it. So it's an extra stimulation above what is going on endogenously.
Okay, they didn't keep in mind, right?
That's a good point. Yeah.
Yeah. So what in the first figure and the first sort of set of experiments basically show that the endogenous CCR5 expression increases with learning, but it's pretty delayed. So the peak of the expression happens 6 to 12 hours after learning, but the expression is also in the cells that were active during the learning experience. So presumably the actual memory circuit.
Yeah, this is really cool because if you in neuroscience, we often talk about this thing called an immediate early gene, which Jason would know a lot about because ARC is one of them. And this is a thing where if you stimulate a neuron, then very quickly, within minutes to an hour, the neuron will turn on a bunch of genes because it's been stimulated recently and it has to process it, to change plasticity and stuff. But this particular CCR5 seems to be a late response gene because you stimulate a neuron and it doesn't immediately turn on. There's a huge delay of hours and then it slowly turns on. So how that works, it's kind of interesting to think about. Yeah,
and there's sort of a lot of gene expression changes that happen after training, and there's definitely a couple of waves. So there's the first wave, as you say, the IAGs, which are really tightly linked to the training. That happen within 5 to 10 minutes after the expression goes up. And then there are these delayed genes that are downstream of many of these first wave genes, because those first wave genes are the transcription factors like CREB. And another thing that's sort of interesting and to bring up, right off the bat is how do the neurons in the hippocampus know when to get active or are actually included in the memory? And so there was a lot of previous work that was done by Alcina Silva's lab initially, and then Sheena Jocelyn, showing that the excitability of individual neurons dictates whether they get included in the memory. So by excitability, I mean we've sort of, this is again neuroscience 101, but neurons have a resting membrane potential, which is usually pretty fixed. And that resting membrane potential dictates whether the neuron will fire an action potential. So it's negative. It's hyperpolarized, and the level of that resting membrane potential can fluctuate. And if it's higher, it means the threshold to induce an action potential becomes easier. So it's sort of more active. So basically, some cells, some neurons, for whatever reason, or perhaps based on their prior history of activation, have a different resting membrane potential, and that's what we're talking about when you say excitability. And so we'll revisit this, but basically it suggests that there's a competition for neurons to be included in the memory. And one big question was, well, that excitability, is there a way to control the excitability so that it goes down so that you could prevent those neurons from being included in the next memory, the next encoding event? So we'll revisit that idea. But for here, the first thing to sort of figure out is how did they design a behavioral paradigm to test the linking of two memories. And so what they've got is two contexts instead of just one. So context A, which presumably has some specific cues, and then a completely different context B, which has other cues. And so those two contexts should be different enough that the mouse can distinguish the two different contexts. And what they'll do is they'll expose the mouse to context A, and then they'll have a delay of either 5 hours or a day or 7 days between exposure to context A and exposure to context B. And the other thing they do is after they've exposed the mouse to those two different contexts, then they'll give a shock in context B with a delay, and then a few days later they'll also then put the mouse in context A. And so then the question will be, if the period of time is short enough between the exposure to context A and context B, will the mouse will link the context B to context A when it's shocked? Does that make sense? So, it's sort of like saying, okay, I went into house A and nothing happened to me. And then 5 hours later, I went to another house, and I got a shock in that house. And because the first house was fresh in my memory, I kind of now associate that the two houses together. And when I get a shock, or because I got a shock in the second house, I don't like the first house as well. And so what they do find, sorry,
yeah, that makes sense.
Yeah,
and it kind of makes sense in terms of like if I'm like in the neighborhood and then I get mugged, then it's likely that neighborhood close by I don't want to go back, because you don't know what will happen. Something like that.
Right. That seems almost like the basis of anxiety, or related to anxiety, because then you start to create these spurious relationships between, some innocuous environment and an environment that is actually relevant to whatever adverse event happened.
Yeah, and it is a tight balance between, overgeneralizing. So anxiety, you could say, is you just generalize the context you got shocked to everything, and that's bad, right? Because then you're constantly feeling like you're anxious, versus being able to discriminate the two different houses and saying, oh, well, actually, I was only shocked in the second house, not the first. And that seems to be, I mean, in the sort of normal way that we sort of encode our memories, time delimited. So within 5 hours, and they show in this fear conditioning paradigm, if you link the context A and B within a couple of hours, 5 hours, there's association, there's a link between them. But if you go past 5 hours, so a day or 7 days, that temporal link is not there. And so then the mouse thinks that the two different contexts are separate.
How do you, I mean, the house metaphor is fine for people, but a human could tell if it's a different house right off the bat. So how do you make the context so the mouse knows that they're different?
I mean, the sensory cues, so usually they change the visual cue, they change the smell. Usually smells are pretty strong. So for rodents, sense of smell is important. There's also contextual change. You can change some of the interesting things you can do to change the grading, the bottom of the floor that the mouse is on can be, these
different materials
squares or big squares, or so you can change the pattern and texture of the actual surface as well.
But I wonder if the mouse can still. So these researchers found that when you re-expose the mouse to context B, which the mouse has previously been shocked in, the mouse obviously freezes. But if you put the mouse back in context A that it previously had not been shocked in, it also freezes as long as A and B are quite close together in time previously. But the mouse surely still can tell the difference between A and B, perhaps.
Well, I mean, I think that's where even in humans, right? Some of this memory stuff that we're talking about is consciously aware. You're consciously aware of, like, yes, I was in two different houses. They weren't exactly the same. But in terms of sort of this overall emotional context to the response to the house, sometimes we feel uneasy or we feel, scared for no apparent reason. And perhaps this is similar. I mean, we're obviously talking about a mouse, but the mouse is probably thinking, I'm not quite sure if I am in context A or B, which one, got shocked, because I seem to recall that there was sort of a link between the two.
Well, it's kind of like we've talked about before. If you eat a sandwich and then for some other reason you get sick very close, and this doesn't actually even involve anything, I think, conscious. So it's working at a deeper level, but you create an association between that food and being sick, so then you can't eat. It's like me and Krispy Kreme donuts, I had a Krispy Kreme donut once and then got sick for some other reason afterwards, and now just thinking about Krispy Kreme donuts makes me nauseous. So, but yeah, it seems kind of like a similar, you may not have to be conscious of it.
Right.
For those types of associations to happen.
I assume whatever cues you pick, you just have to do the control first to make sure that the mice, well, what do? Can distinguish them, but that you don't want them to distinguish it. I mean, is the idea is you put them in a cage and there's a red ball in one cage and not in the other, and that would be enough because they say, oh, I'm in a cage and the ball is there, but I'm still scared. Is that?
Yeah. So the controls are important for sure to say, okay, what is the mouse actually experiencing? So for example, in the second experiment here, where they're trying to manipulate the CCL receptor itself, they throw in a novel context, so a context that the mouse has never seen before, and that's like the baseline control. Because you could just have as Vivian said, you could have an anxious mouse, and the mouse is going to stop, is going to freeze in anything because now it's anxious. And so they show that if you put the mouse in a new test environment, they don't freeze, so that's good. And then when they put them back in the context B, the context they actually got the shock, they do still get they still freeze. So they remember that for sure they got shocked in context B. The part that's sort of wishy-washy for them is context A. And so, in fact, when they do put in, here they've injected Ccr5, which is the ligand to the receptor, directly into the brain of the hippocampus. When they do that after the learning or during this, period, this 5-hour period between the context A and context B exposure, there's less freezing now in context A. In this paradigm, so there's now less of a link between context A and context B. Although it's
but the degree of freezing in B is equal to that of before without Ccr5?
So the amount of freezing to B is equal. So this did not interfere with the actual learning of the original association. So the shock association is not impaired, but the linking to context A is decreased. And at least that's how they interpret the experiment. So that's one way that they've tried to then, activate Ccr5. But in this next experiment, they injected a virus that expresses this light activated version of Ccr5. And then they did the same thing. So expose the mouse to context A, but then during the 5-hour period between context A and B, they shone a light into the hippocampus, which would then activate Ccr5. And again, when they do that, they see less linking, so that the mice don't freeze in context A even after 5-hour gap. But that activation did not impair just the actual association of the shock with context B. So far, so good. So then in the next experiment, now they want to inhibit Ccr5 activity with the idea that then if you had a longer gap between the exposure of context A and B, now maybe you could still link them, where you did not see that in the original experiment. So here, that the way they inhibit the Ccr5 is they use an RNAi approach. So again, they just put in a lentivirus or a AAV virus directly into the brain and express something that will knock down the expression or prevent the expression of Ccr5 during learning. And so in this paradigm now, they do see that with a 2-day gap, there's a link. So the mice show a significant response or fear response in context A. But if you do look, what's interesting is that if you look at the actual numbers here, it's it's significant, but there's a lot of variability, and some mice clearly remember and some don't. But that's just to say that there is a significant difference between a novel context and context A.
Yeah, maybe the mice that didn't learn ended up recruiting the neuronal ensembles that didn't have these knockdown.
Yeah, no, and these are not oftentimes this approach is not effective at knocking down everything. So then the final experiment in this stream of this particular kind of study is that they used now a Ccr5 knockout mouse. So this is a mouse where they've genetically removed the Ccr5 gene. And these mice also have this now linking or increased fear response in context A, now even out to 7 days, when you have that gap between exposure.
Which is nuts.
Yeah.
These are the CRISPR babies group. But like it's crazy that you can have a 7-day gap between context A and B, and then you have an unpleasant experience in context B, and you still attribute all the way back to context A, and presumably everything in between.
Yeah, and that's scary, right? So I think going back to, again, Vivian's observation there about anxiety, one could predict that any sort of traumatic event in the animal's history could be then generalized to a whole bunch of things that happen to that mouse around the same period of time, even beyond 5 hours. Like if you're talking about 7 days, that's a lot of things that can happen.
Yeah, and think about it in human years, right? That's like decades or something.
That's why CCR5 is important. It keeps the memory window short so that you survive as a species. You don't have debilitating associations, right?
Right, exactly.
Jason, I maybe missed this. Did you say that their Ccr5 knockout was, so I assume it was directly like in neurons only?
I think it's global.
This was a full knockout, but that is a good point about in the other experiments where they were expressing either RNAi or that light-activated CCR5 variant, they're using promoters that only express them in neurons.
Right. And it is like you were saying, it's an injection. And I know even, so that's presumably more targeted, although it does, can spread pretty extensively. But, and it's a congenital, like, so from the moment that the animal was born, it's gone, or was it inducible? Okay.
Yeah, no, it looks like it's a full germline knockout. But that's a good point because there is expression of CCR5 in microglia. And so most of their experiments here are manipulating neurons, not microglia. Okay, so they've got their sort of expected phenotypes based on the hypothesis, but now they want to sort of get at the mechanism of how CCR5 activity could really regulate neuronal properties to actually then get at how this is controlling the linking of these memories. So this goes back to previous publications that showed that if you activate CCR5, you change the excitability of those neurons. And so they confirm this by doing electrophysiology from the hippocampal slices. And indeed, if you treat the slice with CCL5, so the ligand again, that does now lower the excitability of the animal of the cells, so that you can't. It basically becomes harder to induce an action potential. And yep.
Sorry, I was just maybe just to be absolutely clear, because the story can be a bit difficult to wrap your head around, but I thought I'd give a really quick summary, which is when a neuron has been recently recruited, for example, if it's been put in context A, the neurons that get recruited there becomes transiently, not transiently, but for a certain time window afterwards, it becomes more excitable. So those neurons, they are kind of like excited kids in a class. They would raise their hands up to be, to answer the next question because they're that much more excitable. But what this CCR5 does is it actually tones down that excitability. So it kind of, but the difference is that this CCR5 only turns on quite a bit later after the initial rate increase in excitability. So the neuron becomes more excitable for a short time period afterwards so that it gets more easily recruited, and therefore you can link the memory together. And then after a time delay, this CCR5 turns on and it kind of fences off those neurons from further participation in neuronal ensemble. And it does so by inhibiting those neurons' excitability. So kind of on switch at the beginning, and then CCR5 comes on, and then off switch to turn those neurons off and fence them off. That's kind of how I think about it.
How deep do they go into the mechanism in this paper? Because I'm thinking, the signaling downstream of CCR5 could end in changing gene transcription, or, for example, insertion of different kinds of receptors at the surface. And when you have changes inside excitability, that's reflected in AMPA receptor, occupancy in different at the synapse, right? So
or potassium channels.
Potassium channel might be one.
Well, I'm also thinking NMDA receptors. There's like a NR3, or I don't know. This is 10 years ago I learned about this, but the idea of metaplasticity. So just changing the composition of an NMDA receptor can then, can regulate how plastic synaptic plasticity is in a given cell.
Yeah. So I would say they didn't delve into that kind of mechanism of what CCR5 is actually doing in this paper, but they do show that the ability to induce an action potential is low. It's it's a potassium channel or some sort of CCR5
is a G protein coupled receptors, in case you're wondering. So it does things via G protein, and I think it's GI coupled, so that would allow it to couple with G protein regulated potassium channels and stuff.
And then,
but apparently also it can be GQ coupled. It's a weird thing.
The previous paper that they reference did show that CCR5 activity blocks CREB activation, and so it's presumably some sort of slower change in transcription, and then a receptor that's important for excitability. But your summary, Tim, was right on. So basically, we think that CREB activation, so this gene that gets turned on with learning, increases excitability, and that helps that particular neuron get incorporated into the memory. And at some point, that excitability has to decrease so that it's not, included in the next memory, and that what they think CCR5 is doing, because it's delayed. And so by the time that it gets fully on, it's a couple of hours after learning, and then the excitability of that cell goes down. So they show that in a couple of ways, that turning down the excitability does end up preventing it from being that neuron being incorporated into the next memory. So they can label the cells that are active during the context A exposure versus the context B exposure. And so if there's no overlap between those two, then you'd expect that there's probably not part of the same engram or memory. But if they are overlapping, then there's probably some commonality between the two. And so they show that after 5 hours, there's a lot of overlap normally. And if you look at 7 days, there's not as much overlap, right? So the two circuits are quite different. But in the Ccr5 knockout, that difference in overlap between 5 hours and 7 days is not significant. So suggesting that, again, the mechanism is probably that Ccr5 is lowering the excitability, and so that prevents that neuron from being incorporated into the next memory.
Yeah, this is the calcium imaging study you're talking about, where they look at like a live mouse with a microscope looking into the hippocampus and which neuron is actually firing in terms of light. They would blink.
Right.
It's interesting that context A, like in a mouse, in a wild type mouse, context A and B, if they are very close together, they activate the same ensemble of hippocampal neurons. Because I think people also find that in terms of place cells. So the hippocampus has this play cells that would fire if you put a mouse in an environment. These play cells would consistently fire at the same place within the environment. And I think people have found that if you put a mouse in environment A, you map out where the play cell fire, and then you put the same mouse in environment B, the same play cells actually get recruited. They just fire at a different location, right? So it is still the same ensemble.
Right.
They just spatial remapping. But it sounds like if you take the mouse and then wait 7 days and put it in a new location, you actually get completely different ensembles.
Right.
Firing. Yeah. I don't know whether that's been found or not. I think these guys might be the first. But they don't talk about play cell at all in this paper.
I would say, so this is an expected finding. I think what's new here is they are using a sort of functional way of measuring activity. And it's pretty cool. So this group is one of the pioneers in these mini scopes. These are like these small microscopes that you can implant on top of the mouse's head. And it's able to image into the hippocampus. So there's like a little lens that drops down. And these mice
it's insane.
I am already thinking, how can I use this?
You literally have neurons that blink on and off like a Christmas, literally blink on and off like a Christmas tree, and you're seeing into them as the mouse is moving.
No, it's pretty cool. And I actually give them kudos as well, because there's a couple of variants of this microscope, this Grin lens microscope, and this group has made these mini scopes freely available in terms of the schematics and being able to build them for much cheaper than commercially.
Yeah, guess how much the one that you can get from a company who would build it for you and come and troubleshoot. Guess how much that is, because they recently sent a rep to us.
Is it over $100,000?
You got it.
Yeah.
Oh, really?
It's as expensive as a house.
Oh, well, houses these days are,
Oh, sorry. Yeah. Okay.
It's quite a bit. That's like a shack somewhere in like rural America. What did you call them again?
The mini scopes.
Okay.
So
definitely going to look that up.
Do people without CCR5 have longer memory linking issues? Do we know?
Yeah, I think this is, because this just came out. So now the implication is, and so in fact, actually it leads into their last experiment, because if, all of this wasn't enough, then they also wanted to throw in some sort of translational aspects to humans. And they wanted to link this to aging. So in humans, there's this observation that Ccr5 and CCL5 expression increases in immune cells with age. And so the prediction there is, if that is also the case in the brain, then they could be age-related decreases in linking memories in humans. And so they did this in aged mice. So these are now 16 to 18 months old, which in lab mice, this is actually, I would say, even older than middle-aged. They say middle-aged. I would say this is pretty old. And so what they do find is that in these older mice, CCR5 expression is increased. And this linking effect that they see even in 5 hours in young mice is absent in these old mice. But if they looked at the similar aged Ccr5 knockout mice, those mice now had normal linking at 5 hours. So finally, to your point, Vincent, there are drugs, as was discussed, that can inhibit Ccr5. So this is an FDA-approved antagonist for HIV treatment in humans. And they took the same drug and infused it into the hippocampus of these old mice. And so this is, what is it called? Maraviroc? Mm-hmm. Giving this drug to the mice now restored this linking effect for 5 hours. So pretty interesting that this HIV drug now could actually affect memory.
I'm amazed, because the drug was selected to block HIV attachment. And so you would think it's a different part of the protein that's functional in memory linking, right? So they're pretty lucky that it worked.
Yeah. And so I think the implications for humans are sort of really interesting, and presumably there's data out there for people that have been taking these drugs, I'm guessing you could try and do some studies on them for memory.
Oh, that's a good point, because you have to take it your whole life, and so you could have noticeable effects. But we also have a lot of people that don't have CCR5, right?
Yeah.
Natural. And I wonder, you would have thought that if they had some memory linking issue, we would have known.
Right. I mean, and these may be subtle, but what they do say at some point in this whole paper, of course, is that this is not the only mechanism to link memories, and there's presumably other molecular mechanisms as well. So I think it's sort of interesting that there's because the evolution uses these receptors in different ways in different parts of the body. Even though you think you may have a clean drug that's only affecting the immune system, you may not know what else it could be affecting.
The CCR5. Go ahead, Tim.
Go ahead, you first.
I was gonna ask: is this drug, the CCR5 antagonist, able to cross the blood-brain barrier? And would it be helpful? And is it something that's prescribed for somebody who's developed the HIV-associated neurodegeneration?
Actually, that's a good point. I don't know.
Would it really be?
I don't know.
I presumably probably not that effective at getting into the blood, getting into the brain, most because there was no sort of reason for them to develop a drug that couldn't get into the brain, although we know that the brain is a big reservoir, latent reservoir for HIV infections. And so maybe if you could get a drug in there, it would be better. But
it does get into, the drug does get into brain.
Okay.
Yep.
Interesting.
So they
most
looked
of
at
the
it
time
because
it's the opposite.
because of the brain, associated HIV symptoms. But so you could treat, but it could be redundant in people, right? There may be another way of dealing with the memory linking, and that's why people who lack CCR5 don't have any obvious issues. Or maybe they just haven't been studied. As you said, it might be subtle. It's a good question. I
had two questions. So I think at the beginning, Jason, you mentioned episodic memory. How does that relate to explicit and implicit memory? Because I think that would be difficult. In humans, I think you can differentiate between explicit and. So I feel like explicit and episodic frequently would go together, but maybe not. It wouldn't exclude necessarily implicit memory. So that's something that we as humans can appreciate and we all understand. Maybe we don't know the words for it, but then in mice, how can you possibly ask a mouse if it knows where it's learned something?
Yeah.
I mean, I guess that's kind of like what you were, you could kind of infer that from the experiments that the behavioral assays they did here. But
right. Yeah. No.
So I'm just wondering.
Those are debates that we have in the memory field. Is we think we're modeling episodic and explicit memories, but you can't really ask the mouse exactly what they're experiencing. So you sort of have to infer it. But you can dissociate mechanisms between implicit and explicit, even in mice. So, for example, motor learning tasks where there's tasks where you can train the animal to grasp a piece of food outside of its normal range, and so there's a movement that's required. Some of the mechanism, so you can take out the hippocampus, and that kind of memory is fine. You take out the cerebellum, they can't remember. So some of the mechanisms that are conserved there with humans, in that even the sort of classic HM patient who had hippocampal lesions could still remember playing a piano. So you ask HM, can you play the piano? You say, no, I can't consciously. I don't know how to play the piano. But you put him in front of the piano, he can automatically play it. So
he can, not only can he remember how to move his fingers, he remember the tune as well.
Right.
This kind of reminds me of my second question, which is, or I don't know, comment, rambling thought, which is, there was a series of lectures that were part of my graduate school, like first year courses, about studying, like how to study, how would one study to like really leverage your your memory system so that to improve recall and things like that. And, so there was this idea that you should study and then you should sleep, but not too much. And then the next day you'll remember it better. And I've also heard, you should study the material in multiple different places or in multiple different orders. And I'm like, with respect to studying in multiple different places, are you recruiting multiple, I don't know how to use the words, but multiple engrams that lead to the same memory, or are you actually dissociating the memory from either location? Do you know what I mean?
Well, so what's definitely true is that recall in the same place that you learned is much more easy to do that. So if you were to learn the material in different contexts, you're sort of guaranteeing that maybe the recall of that material will be better because you've learned it in different contexts.
You've just increased your chances of remembering it by broadening the number of places that. Yeah,
exactly. Okay. And then sleep for sure. So my best advice for people trying to remember for learning for an exam, for example, don't cram the last minute and forget to sleep, because then you're definitely not going to remember it. And you definitely need that consolidation during sleep to really get the best learning. The other thing that's common from flies even to humans is this idea of space training. So you train once and then you have a bit of a gap and then you train again. And so that having that spaced training can really help as well. And that's partly because I think that you need that sleep in between to guarantee consolidation.
I got a question, which these researchers didn't answer in this paper, which is, if I put the mouse in context A and I activate those engrams, those ensembles, and then I wait 6 hours, then the Ccr5 expression goes up to fence off those ensembles from being recruited again for the my next experience. But what if I present the mouse, or I am in context A, and then a few hours later, I go back to context A? Do I now delay the Ccr5 from being expressed so that I just keep activating the same ensemble over and over again? And would that explain why during the pandemic, I have all my experience rolled into a single mush?
Yeah. Jelly roll of malaise.
Exactly.
There is evidence for sort of transcriptional inhibition. So the more times you come back to the same context, the less excitable those cells are, because they're just like, oh, well, you're in the same place. Why should we care? So repeated activation of the same cells, or the repeated experiences, leads to less activation of the same cells.
Okay. Oh, so that would also explain why I don't remember things in the pandemic. My brain just turned off.
Why novel experiences are so good for the brain, because now your brain is like, I should care about something. So, going on vacation, doing anything new that you've never experienced before is good for you.
You see, Tim, it's either this that we're talking about, or I don't know if you ever got COVID, but maybe you have.
Not yet.
Not yet?
Would you believe it?
Amazing.
Yeah.
Is it because you just stay inside?
Because I'm just in the lab all the time, exactly.
You're so dedicated. And Jason, you've escaped
as far
as
as
well?
I know, I've not got it, but I've traveled, so I guess I've just been lucky. But
you may have it or have had it, but you don't know because we're totally asymptomatic. Yeah.
Totally.
Vincent, before we wrap up, I was just wondering, I think you said a while back, like that we're going to learn a lot about human immunity from this virus because it impacts different people so differently. Do you have any quick updates about, do we understand why Jason and Tim, well, we know Tim, he doesn't go anywhere, right? So we'll take him out, but Jason travels a lot. Why has he gotten it? Or he not gotten it? But others who travel do, assuming that they both mask and follow the same protocols.
So mostly, go ahead.
I was going to say something interesting about that is that if you look at infectivity rates in Africa, it's much lower than outside of Africa. And so I grew up in South Africa, and there's this idea that maybe the vaccination regimens or whatever we got as kids has been beneficial, but I don't know if that's panned out.
Mostly we have understood severe COVID, why some people get very sick, right? Less so why people don't get sick at all. But, for severe COVID, there are a lot of polymorphisms in various, mostly innate immune genes that regulate. And if you don't make a really good innate response, you're going to have severe COVID. It's really important for controlling the initial infection and also for getting a good adaptive response because the innate and adaptives collaborate extensively. And so by extension, it could be that people who don't have symptoms have a really good innate response and they clear the infection before it gets going very, very much, right? But it's not clear. I think it's going to take some time before we understand these things. So my comment was to say, yes, but it's also going to take a long time because, as you know, these issues are not sorted out quickly, even though during a pandemic, people want answers quickly. You're just not going to get them. So,
Okay, final question for the paper, which is, in this paper, we found that an immune-related gene affects our nervous system's memory. But in your body, there are famously two different systems that have memory. One is your nervous system, and the other is your immune system. So I'm wondering, do you need a way of fencing off your immune systems so that you have different memories that are separate? And does this get involved? So, for example, if I go to a park this afternoon, if I go to Central Park and I get exposed to like a weird new pollen, and then later on tonight, I go to a concert and I get a horrible flu. Is there a way for my immune system to separate my afternoon experience from my late night escapade and getting the flu? Yeah, I don't know. It's weird to think about.
Well, your immune system can do it. That's no problem.
Okay. Is there a regulation of this process so that your timing window?
No, it's just that you have different sets of B cells responding to different antigens, right?
Right.
And they will independently become memory B cells. Every B cell recognizes one epitope. And if you're exposed to that, those B cells will amplify and become memory B cells and stay with you forever. Or a long time anyway. So
they're all independent. Every antigen is going to be functioning independently.
But my question is, if I am exposed to a new pollen in the afternoon, and if they're still presented by the antigen-presenting cells, and then later on tonight I get a horrible flu, is there a linking of my new pollen to this flu which is a mistake because the pollen is actually harmless?
To a certain extent, because in both cases you have a lot of chemokines and cytokines produced to facilitate the process, and they may overlap. Yeah, right.
Yeah,
but the cells are distinct for sure.
Right.
So your question is by analogy to this memory linking that we've been talking. Yeah, right.
In the immune system, is there a temporal window in the immune system?
I think
does anyone know about it?
Identified a new paradigm shifting set of experiments.
Well, I'm trying to think, why would you want to do that?
For allergies, so that you don't get, there are harmless allergens, harmless outside foreign things that you don't want to make an immune response to.
That's true.
And when you see a pollen and there's no things that triggers an innate immune response, you want to ignore those things. That's what I'm kind of thinking. I don't know. There
is certainly overlap because inflammation is inflammation, right? And so if it's caused by one thing, it will participate. Inflammation has a reason to exist, right? Inflammation helps make a better immune response. So it's going to help. If you have a horrible allergic response, maybe it will help and you get a vaccination, maybe it will help that,
Yeah.
Or vaccination may exacerbate
adjuvants, isn't that what it's called? Adjuvants?
Right.
An adjuvant is,
Right.
that helps immune responses. But, we're talking about different, you get a vaccine and you may get worse influenza because you're in the process of having inflammation by the vaccine, and that's going to contribute to the influenza because most of the signs and symptoms of an infection are immune-based, not actually caused by the virus. So there is an overlap for sure, Jason.
And that was actually the other paper that I was thinking of discussing was a new paper sort of showing how the brain changes its behavioral response to sickness. So how do you get to sickness symptoms like fever and those sorts of different things that can actually influence getting, recovering in response to an infection. But this is not.
It's a shame we didn't do that as well.
You know what?
Well,
we
next
can do
time,
it next month.
And next time.
I want to know what's the lowest organism that still can have a fever? I don't know. We'll find out maybe next month.
They have to make cytokines, right? Because those are what trigger fever, and particular cytokines are known to do that. So you just ask which ones make it.
Make cytokines.
Yeah.
But I think a lot, for example, do cold-blooded animals, do they get fever?
I don't know.
But I think most animals have sickness behaviors that it may not just be, that are not just fevers, but things like, being less energetic, not running around, stuff like that.
Yeah,
Right.
you should. Your next turn, Jason, you should do that paper.
Sure.
Of course, there will be a million others that pop up that are fascinating. Tough choice. All right, that is TWiN 31. You can find the show notes at microbe.tv/TWiN. If you have any questions or comments, you can send them to twin@microbe.tv. And if you enjoy our little chats here, please consider supporting us. You go to microbe.tv/TWiN. No, sorry, microbe.tv/contribute. Jason Shepard is at University of Utah. Thanks, Jason.
Thanks, Vincent.
And also on Twitter, Jason Synaptic, all one word. Tim Chung is at New York University. Thanks, Tim.
Thanks, Vincent. Thanks, Jason, for that paper. It was very fascinating.
And Vivian Morrison is at Tulane University. Thanks, Vivian.
Thanks, guys. I'll echo what Tim said. Really, really, really interesting. We clearly could have stayed here for a lot longer.
Well, hopefully we have whetted the appetites of the listeners to go and learn more. That's what we'd like to do. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.